Yeah, good afternoon, Dr. Lonzarich here, and so this is unit number 3, and this is the second subject. First was the immunology material that I posted on Friday last this past week. This is for the fourth week of 6 in the second half of the semester, and this whole week is devoted to nervous system, and next week is devoted to endocrine system, and the next week after that to reproduction and development. And that will be the last, that's the last unit. So it's 6 weeks, no, 6 weeks, 3 weeks and 9 lectures. All right, so this lecture slideshow is actually 12 slides long. I'm going to try to get through it in one sitting. That's my goal. And you can sit through it in one sitting or watch it in chunks. So animal systems of communication. Well, of course, one of the major distinctions between animals and everything else is the complex systems of communication that exist. Plants have something similar to endocrine system. They have hormones. They don't have a system per se. They do communicate chemically. Animals are the only organisms on this planet that have nervous systems. And one of the objectives that is set out for you is to think of this comparatively, like what kinds of nervous systems exist. But the focus over the week will be on general features of nervous systems, functions of nervous systems, how the nervous system's communication works, and then I was going to finish off with sensory systems, but I'm only going to really do one example on the back end of this. All right, so one of the things that you want to know is how the endocrine systems and the nervous systems are compare, I should say. How are they different? How are they the same? They are complementary systems. They do interact. One of the questions I will definitely ask you is to think about how these two systems interact and in what situations. I'm going to just leave this here for you to digest. You are responsible for its contents. Moving to the next slide. So the focus here from here after for the rest of the week is nervous systems, and as I said, one of the things I want you to know is how nervous systems differ among groups. But this is my general overview for today. So nervous systems patterns among groups, nervous systems functions, and neurons in most animals, except for sponges. They don't have neurons. So what do I want to have you learn here? This is it right there, about how different groups compare. It's only in the chordates that you have a dorsal nerve cord. There are a lot of animals that have brains, but the spinal cords, if you want to call them, that exist in invertebrates are ventral in their position, and then the chordates have brains, dorsal cords, and then the jellyfish and the sea stars, echinoderms, have nervous systems without brains, and the nervous system of the jellyfish is called the nerve net. I am not going to spend any more time talking about it, but I will have you know that the nerve net in a jellyfish is unique in one additional respect. There's no brain. Nerve net communication is multidirectional, and that is important because in every other organism, nervous system communication is unidirectional, and we'll talk more about that. Nervous system functions, sensing the environment and responding to stimuli. Those are two very broad general functions that you'll definitely want to remember. And then we will get to neurons too. Nervous system overview continuing. In the vertebrates, there are two systems. and I'm going to guess most of you have been through this before. Central nervous system, which is composed of the brain and spinal cord, contains relay neurons or interneurons, which we will talk about. And then the peripheral nervous system, which is composed of the nerves and neurons outside of the spinal cord and brain. They contain sensory neurons and motor neurons. The peripheral nervous system and central nervous system differ in location and type of nervous system components. There are further subdivisions. There's the somatic component, which is involved in voluntary activities, and the autonomic components, which are involved in involuntary activities. And I will leave to you to give examples for these two. Cells of the nervous system, well, certainly the neurons are the principal player. Most of the cells that we will focus our attention on are going to be neurons. There are also glial cells, which are support functions for neurons. For example, insulating axons of neurons is a function of glial cells. Nerves are bundles of neurons. That's important to remember. Nerves are not the same thing as neurons. All right, moving on. All right, continuing. So now we're focused attention on neurons. This is all about organization, right? Neurons are a fundamental unit of the nervous system. That is to say that when we focus on nervous system function, we typically focus on neurons. Generally, when we talk about the structures of neurons, distinctions that exist in neurons compared to everything else, they have dendrite components, which are these end pieces here that are branching or root-like. Then there's a cell body, and then there's an axon, and then there is a synapse. And the synapse is the connection that exists between one neuron and another cell. That cell could be a neuron, could be a muscle cell, could be a gland cell. And there are other things that I want you to remember about this, like, for example, this here, right, this piece that's noted by this word axon hillock is an important position in a cell, which we will talk later about. Then when two cells are connecting to each other, often you will hear people talk about presynaptic cells and postsynaptic cells. That's another thing you want to remember here. Important thing that you will also want to remember is that signaling within neurons is electrical. That is to say, within, right? Inside of neurons, it's electrical. And there are two types of electrical signals. There are graded potentials, or I should say, signals created by graded potentials, and there are signals created by action potentials. And we will spend more time talking about those. It's a very important part of this whole story is to know the difference between graded and action potentials. You'll often hear people say membrane potentials, those are graded potentials. Information flow is unidirectional except for in the jellyfish. So information flow is always in this direction, all right, which of course I'm sure many of you already do. Types of neurons. Neurons are fundamental units of nervous systems. I already said that. So there are sensory neurons, there are interneurons, there are motor neurons. And yeah, this is all good stuff, right? So sensory neurons are on the front end. They are receiving information from sensory cells. So cells in the eye, cells in the ears, cells in the mouth. And they are signaling, they're transducing the signal, the nature of the signal coming from those sources. It could be chemical, could be mechanical, could be visual, electrical, and transducing them and sending a signal down the cell, this sensory neuron. And that sensory neuron will communicate with interneurons, which typically are found in the central nervous system. In fact, maybe always found in the central nervous system. And then the interneuron is think of this as a processing neuron. So when interneurons are together, we think of it as a processing center. And then those interneurons will communicate with the next set of cells that are found in the central nervous system, which are motor neurons. And motor neurons are in the peripheral and central nervous system. They transmit information to target cells, and those target cells could be muscles. So it would be going like this, to this, to this, to this. And so we can think of glands and muscles as being targets, and they're highly branched axons. one of the things that's really important for you to remember here too is that there are certainly, when you look at axons and dendrites and you look at neurons in general, you see how branched they are, but it's also important here to remember that um, it's not just one to one to one, right? It's potentially thousands of connections that exist between one cell and other cells. Um, and so this is a highly complicated network of branching and connections that exist in these neurons. Information processing. This is a summary of what I just said. This is a simplified view of things. So sensory cells detect cues. Here are the types of cues that could be picked up by the body. Sensory neurons send a signal to the central nervous system. Interneurons pick up that signal. They communicate then to motor neurons that generates a response. So here's an illustration or an example of something like that. All right, now we get into some of the harder stuff. And I've already said that inside neurons, signaling is electrical. And, you know, if I'm teaching a biology 105 class, I would leave it at that. It's like electrical and communication. It's important to remember that communication between neurons is not electrical. It's chemical. It's neurotransmitters communicating with other cells and inducing a response in those downstream cells. We will talk about that. But for here, focus is entirely on the electrical signaling takes place within the cells. So we're talking about two kinds of potentials, well actually three. We're going to be talking about membrane potentials, we'll be talking about action potentials, we'll be talking about resting potentials. And so the story is going to be pretty heavy. There are videos that I have made available to you. Some of you have heard about this already before, so this is a bit of a refresher. And so when you think about electrical signaling inside cells, there are a lot of questions that should be coming to mind. So first, of course, is how do you get electrical signaling? Well, the electrical signaling is not going to involve electrons. That is to say, we're not talking about electrons moving like in copper wire in an electrical system in your house. What is moving, of course, then it begs the question, are ions. And the ions, there are two major players, although there are certainly others that are involved. Potassium and sodium. Actually, calcium is really important too, but not in this story today. Sodium and potassium are the major cations. Chloride and proteins are major anions. And so the first thing that you want to think about when it comes to the electrical conductivity or conductance in a cell is the creation of a resting potential. And of course, you're probably going to ask yourself, well, why does resting potential matter? That's a good test question. And you'll see it in a graph in a moment. So the resting potential is a potential, that is to say a voltage, that is created across the membrane of a resting cell. And that voltage is created by the movement of ions. And those ions are distributed across the membrane. And here is the explanation for how it happens. The ions are distributed across the membrane such that there is a voltage that is created in a resting cell. And when we talk about a resting potential in a human body, human cell, it will typically be reported as this, minus 60 to minus 80 millivolts. So, to summarize, the first thing that you want to know about voltages, or I should say electrical transmission in a cell, is the creation of a resting potential. And of course, again, why the resting potential is important will be evident in a moment. All right, then what happens next? Well, rapid changes in membrane potential create signals that propagate in the neuron. So how does that happen? So here we have action and graded potential. So I talked about three types of potentials. I talked about resting potential, which is created in a resting state by the movement of ions. And then there are action potentials and graded potentials, or sometimes called membrane potentials. Actually, can I correct myself? I think about membrane potentials, and I'm confusing myself. I'm going to get back to you on this. I'm not going to restart this video. Certainly think of action potentials and graded potentials as different. Now I equated membrane potentials with graded potentials earlier, and now I'm not sure that I did that correctly. So let's just discard the equivalency of membrane and graded potentials for the time being, and I'll get back to you if I have to. But we do have action potentials and graded potentials, and both of those are changes in voltage that create something, some response in the cell. The resting potential is a voltage that is at the cell membrane where the cell is at rest, there's nothing happening. All right, so now we talk about these two types of potentials, action and graded potentials. And these are potentials that create something, some change in the voltage. They could create, one could create the other. So graded potentials could lead to action potentials. But graded potentials are not the same thing as by me presenting it here. Clearly, they're not the same thing. So let's talk about the differences. So first, let's talk about what happens for both of these. The ion channels open with the stimulus. So the stimulus could be and is likely chemical in nature. It could be otherwise. It could be pressure-induced change. But when we typically think about this system, we typically think about chemicals. So ion channels open with the stimulus. So it could be chemical or otherwise. The result is a change in the movement of sodium from outside of the cell into the cell. And this is a result of sodium channels opening in the cell membrane. We call this a depolarization. And then subsequently, there is a hyperpolarization, which is the result of potassium channels opening and the diffusion of potassium out to the cell. So here's the example. So depolarization is a rise in the voltage, and hyperpolarization is a descent back to a level below the resting potential, as you can see here. And we'll talk about why hyperpolarization is important as we move forward. So differences in potentials. Well, a few things. One is graded potentials are typically found on, and maybe always found on, the dendrites. And they're ligand-gated channels. And what does that mean? They're chemically generated responses or stimuli. Vary with strength of stimulus, and they decay with time and distance. So when a ligand-gated channel is opened, more ligand-gated channels that are open will generate greater or differences in strength of the stimulus. And that stimulus will only have a localized effect. And so as the farther you go away from the stimulus, the less strong the effect is. And so it decays with distance and also with time. Action potentials are not chemically induced stimuli, or not induced by chemical stimuli, but rather voltage stimuli. And that means that there's a constant magnitude and self-propagation of these signals. And so they involve these voltage-gated channels. When we talk about action potentials, we're talking about axons. We talk about graded potentials, we're talking about dendrites. And dendrites, when a collection of graded potentials are sufficiently strong enough, they can induce an action potential. But until that threshold is reached with graded potentials, there is no action potential. So graded potentials induce action potentials. Graded potentials need to be strong enough to induce an action potential, Once an action potential is started, it's like a dominoes. It'll just continue. And where does it continue? It continues all the way down from the axon hillock to the base of the axon, or the axon terminal. And that will lead to something else, which we will talk about later. So here's graded potentials, which have to be strong enough to induce a action potential, which begins at the axon hillock. And then once the action potential is started here, no more signals need to be produced. It will propagate all the way down, like I said, like a domino, collection of dominoes falling. All right. Graded potentials travel from dendrites through the cell body, right? Action potentials only occur if graded potential exceeds the threshold potential. You should know why. And it has to do with the voltage-gated channels. Voltage-gated channels are those channels that are opened in the axon. And you have to have a strong enough voltage in order for those channels to open. So here's the threshold. If the threshold for the voltage-gated channels does not get met, no voltage channels get open. So these would be failed initiations of graded potentials that didn't get strong enough, and they died out as they went along the dendrites to the cell body. But if there are strong enough ligand-gated channel stimuli that elevate the voltage to this threshold, then you get a propagation of the action potential. Sodium channels open, depolarization, then you get a repolarization, and then you also get a hyperpolarization. Action potentials, again, are self-propagating. They depend on two types of voltage-gated channels opening and closing. What are those? There are sodium channels first and potassium channels second. So here we have the numbers aligning with the functions. I think numbers aligning with the functions. Threshold potential is exceeded. So actually, that's the first step, and that would be here. Sodium channels open, and this 3 is depolarization. Potassium channels open, and sodium channels close. That's the repolarization here, and then a hyperpolarization here. And those hyperpolarization are important because they create what's called a refractory period. There are two kinds of refractory periods. I'm not interested in discussing the two types here, but the refractory periods generally are conditions of the potential at the membrane which will prevent the movement of action potentials in the opposite direction. So if you think about how these action potentials are propagating, it's along this axon all the way down to the axon terminal, and it's always in that direction. And so one action potential generates another action potential. So it's one action potential goes downstream to another part of the membrane, which then will trigger another action potential, and then it will continue to trigger other action potentials all the way to the axon terminal. It can't go backwards, and that's because the voltage is too low for it to initiate the opening of sodium channels in the opposite direction. So this is one way to ensure the efficient movement of information from the axon hillock to the axon terminal. There are 10 to 10 billion neurons in the brain, and each neuron receives signals from many other neurons. Each neuron will send signals to many other neurons, and each neuron can have up to 10,000 synaptic connections. And here's something pretty remarkable. Each synapse may have several neurotransmitters. So you can imagine just how complex and complicated the story is when it comes to communication. So let's see if I think this might be my last slide. How signals propagate. Signal from presynaptic cell to postsynaptic cell. So we've got graded potentials, action potentials, and then once we get down to the axon terminal, we're going to have a synaptic connection between the presynaptic cell and something called the postsynaptic cell, which could be a gland cell, could be a blood vessel, could be a muscle cell. And the communication that takes place there is going to be chemical, and they will induce graded potentials in the postsynaptic cells. Those graded potentials could be excitatory, could be inhibitory, and as I said before, you have to get a lot of graded potentials in order to generate the sufficient threshold or sufficient voltage change to induce an action potential. And then action potential and an axon does what? It travels down to another cell connection at a synapse. One last thing I want to say, I don't think I've shared it yet. You see these things here, these bud-like structures. That's myelin. Humans, vertebrates, have myelin-covered axons. And the reason why that's true is because it speeds electrical transmission. So when an action potential starts, the next gates that open are here. And then the next gates that open are here. So they call it saltatory transmission. So it's jumping from gap to gap. And that actually generates a greater speed of transmission, which makes this axon much more efficient. Squid, which is where this whole story was first explored, have really large diameter axons, no myelin sheaths, no covers. And they can get speed of transmission because their axons are large diameter. And that is one way to speed the movement of things. But in animals like us, we don't have large axons. So we have to speed transmission by generating these sheaths that then create the saltatory conduction of information. All right, that's it. That was a long lecture. Sorry.